Hello, this is Martin Horvath, and I'm preparing this video for students of Biology 3510 at the University of Utah. And today, the focus of this video is understanding the biological purpose of MutY and also its chemical mechanism. So MutY is an example of so-called base excision repair enzyme. It acts on DNA to remove base from the DNA so as to prevent mutations. And we're going to begin the story about why we need MutY by considering a G-C base pair in DNA. And G, of all the bases, is most sensitive to reactive oxygen species, ROS. And it will become oxidized. We can write here OXO-G. And OXO-G is it can base pair with C, but it can also, with a round of replication, base pair with adenine. So the reliability of this template is compromised by this damage on the G base. So this arrow here is really two arrows: attack on the guanine by reactive oxygen species, and then replication, synthesizing a daughter strand across from this ambiguous template, 8-oxo-G. And then if there's another round of replication, now we have a big problem, because this A will template the insertion of a T in the next daughter strand. And then now we have a TA base pair. And that's a completely different piece of information from where we started. We can call this a mutation. This OG 8-oxo-G adenine base pair, we don't call it a mutation. It's not a mutation. It's not permanent yet. There's still a chance of catching this and repairing it. So we call this a lesion, or a site of DNA damage. And what MutY does is it intercepts this, finds this lesion and puts it on the pathway for repair. Here's the 8-oxo-guanine, and it does this by creating an abasic site, a piece of DNA with no base. So the adenine base is getting removed from the DNA by MutY. So we'll write here MutY. And by this abasic site is now the substrate for other enzymes that we can draw in here. There's at least 3 of them. And we can repair this to 8-oxo-G across from C, and then that is a substrate for another enzyme that has similar properties to MutY. In humans, it's called OGG1, and it will remove the OXO-G as a base from the DNA, leaving an abasic site across from C, and then those same enzymes, those general DNA repair enzymes that helped us here, will restore the GC base pair. So what MutY is doing is it's putting it on this repair pathway, and thereby it's preventing the mutation. And how does it do that? Well, we have to consider the chemistry and also the active site for this enzyme, and we'll begin here by drawing out the structure of a piece of DNA, and it's here. Drawing adenine base completely. And this is a structure of DNA in a DNA strand. It's part of double-stranded DNA, And we're not going to show the 8-oxo-G. It's on the partner with the adenine. But this is not just any adenine. It's an adenine that's base paired with oxidized guanine. And what the enzyme provides is a catalytic acid-base group, and this has a name. It's glutamate 43. It's 43 at position 43 in the species that I study. It comes from a bacterium. In humans, this label would be different, but it has humans. The human MutY homolog also has a glutamate at exactly the same place. And one way to think about this acid-base chemistry is that the protonating this N7 nitrogen is going to pull electrons out of the ring, and that's going to pull electrons out of this covalent bond. And it's this bond that we need to break in order to remove the adenine and begin the repair process. So this is an example of acid-base catalysis. And what we get from this first step is a transition state where we're in the process of breaking this bond. We're protonated here. Sorry, maybe it's more appropriate to put this double bond here now. And this is the leaving group, LG. And another way to think about what glutamate 43 is doing as an acid-base catalyst, it's protonating the leaving group, thereby stabilizing the leaving group properties, making it a more favorable situation for this to act as a leaving group. And as we do this, we're accumulating positive charge. The electrons that were in the bond are leaving the carbon, leaving behind a positively charged carbon. And this is a very unstable species. It's called the oxocarbenium ion transition state. So what the enzyme further does is it has a negatively charged group that we'll draw in here. It's there as a permanent part of the active site called asp144. It's an aspartate residue. So aspartate, with its negative charge, helps to stabilize this transition state through electrostatic interactions. And that's a common theme for many enzymes. They provide charged groups or metal ions to help stabilize the excess charge on the transition state. In the textbook, most of those transition states have excess negative charge. Here, in the case of the MutY catalyzed reaction, the transition state has a positively charged carbon. In organic chemistry, you would call such carbon the carbocation. And because there's an extra oxygen adjacent, we now call it the oxocarbenium ion. Still very unstable. But it's not quite as unstable as it would be in solution because of the active site residue aspartate 144. Okay, so we've created we're now going to picture the leaving group diffusing out of the active site, and we're going to bring in the next the substrate for this reaction. Draw out the transition state again here. Now we have a little bit more room to really clearly picture aspartate 144 favorably interacting with the positively charged carbocation, and then we now have glutamate 43 with its negative charge, which also helps stabilize the transition state. But in this case, we're going to use the glutamate 43 catalytic residue again as an acid-base catalyst. So water comes, this is going to be the nucleophile in the final phase of the chemistry, and by taking the proton away from water, we build a much more aggressive hydroxide ion as a nucleophile, which will attack and form the bond with this positively charged carbon. And the final structure then is drawn out here. We have a strand of DNA with no base attached. There's all along there's been this hydrogen at this position. Drawing that hydrogen. So we're back to a stable tetrahedral carbon attached to four things and with no excess charge. This is an example of an SN1 type reaction, meaning the leaving group has completely dissociated from the transition state before the arrival of the nucleophile, and it's an example of electrostatic stabilization of the transition state using aspartate 144, and then we have in two steps, glutamate 43 participates as an acid base catalyst. And we have a name for this final thing. We call it the abasic site. It's also called the AP site, meaning apurinic, apyrimidinic, a piece of DNA with neither a purine nor a pyrimidine attached.